So we left off talking about neurons and their myelin sheaths. Let's go and just do a quick review. So, nervous system, 3 divisions we're going to talk about, right? Sensory, which is input information, that's what's coming to you from my voice. Integration, brain, kind of sorting out the information, pulling files, and then a response, motor. Yes, do we understand this? Yes, we will. So all of these are part of that whole 3 divisions we just mentioned. Central nervous system, brain and spinal cord, this is where the information is all coming to and going out of. Going out to what we call the peripheral nervous system, which consists of sensory input information and output information, motor. Sensory is afferent, motor efferent. The motor division, things going out, is also divided into things that happen conscious, somatic, and things that happen automatic, autonomic. And that division is broken down into things that are sped up and things that are slowed down, sympathetic and parasympathetic. So throughout these chapters, we'll talk about all of these divisions in detail. Glial cells, what are they going to help do? What do glial cells do? This is a what kind of cell? Astrocyte. Then we have a microglia. We also have some ependymal cells and oligodendrocytes. Where are they all found? Where? Central nervous system. Peripheral nervous system, we see Schwann cells. Schwann cells are going to help along with oligodendrocytes in the central nervous system. What do they provide for neurons? Begins with an I. Insulation support. They're going to help impulses go down those axons without leaking out the sides. When I talk about impulses, I'm talking about ions, charged particles, moving through that axon. That change in charge that's going to occur in the cell when a nerve impulse or action potential takes place is going to force out at the end of the axon some very important chemicals. These chemicals are called neurotransmitters, and they're going to cause changes in the cells after these axon terminals. So, we left off with, they don't reach out, they spit out. Now they come in contact with many others, cells, right, so it's not just one, it's many, but the way they communicate is through these chemicals, neurotransmitters, and we're going to see what happens within those cells when the neurotransmitter comes in contact with the next guy in line. So this is an example of Schwann cells and how they wrap themselves around axons in the peripheral nervous system. And we talked a little bit about that looking like, you know, the little toothpaste thing that you get. I have one at home because I'm really cheap and I want to make sure I get all the toothpaste out of the tube. Yes? That's how the oligodendrocytes and the Schwann cells wrap themselves around axons to help insulate them. So, certain toxins, certain chemicals can cause problems with neurotransmission. And that's what they talk about under homeostatic imbalances. So, and by the way, I'm on page 392 as we go along. So the myelin sheath then, in the peripheral nervous system, 392. In the peripheral, no, it's next. But I'm talking talking about the stuff here in page 392. That's all right. The myelin sheath then, peripheral nervous system, Schwann cells are going to create little gaps. Little gaps are called the nodes of Ranvier. And we're going to see how it's a little bit different when nerve impulse passes down the axons of a peripheral nerve versus nerves in the central nervous system, or neurons in the central nervous system. Again, these cells, both of them create the myelin sheath. So when we talk about myelination of cells, we're talking about these cells wrapping themselves around the axons. So the myelination of neurons is actually white in color. So I think we mentioned this a little bit, white and gray matter. When we look at the nervous system, especially the central nervous system, we can actually see a difference in color. When you dissect sheep brains in lab, which is coming after the many weeks of bones, you are going to see a difference in color. White matter is what? Why is it white? Because of the myelin. So myelinated fibers, white matter. Gray matter, unmyelinated fibers, and cell bodies. So we're going to see a distinct difference because of this myelination in the central nervous system. So we have different structural classifications of neurons. And it depends on who they communicate with and how message gets sent down their fibers. In this table, we see some of the different classifications of neurons. We have things called multipolar neurons. They have 3 or more processes in one axon. Then we see a bipolar neuron. Bi means what? 2. Have 2 processes, an axon and a dendrite. And then unipolar neurons have a single short process that emerges from the cell body and divides like a T. All of these different types of neurons will send messages at different rates and be triggered at different points. This allows for them to be either faster or slower in their delivery of message. So, neuron type, multipolar. Multipolar neurons, many poles. And again, we find these in different places in the body, receiving different amounts of information.
You say many poles, you're talking about dendrites?
Yep. If you look at the picture, look at how many dendrites we have feeding into that cell body. These are the most abundant in our bodies, especially in the peripheral nervous system. We want to get as much message as we can to our neurons at any given time. So, yep. So by having many, many dendrites, I can pick up message that's coming from a neuron up here, for example, or some stimulus that's coming from the environment.
Dendrites
Receive information. Dendrites are the receiving end of the neurons. Cell body's kind of where everybody has to pass through, and the send out information part is the axon. Bipolar neurons, not as abundant in the body, but when we talk about sight, second semester, first chapter, we're going to talk about bipolar neurons. So we'll see these kinds of neurons in senses like sight. We also find them in olfactory. What's that? Our sense of smell. Yes. We find them in the inner ear for our sense of hearing. So when we talk about the special senses, we're going to talk about these guys. And then found mainly in the peripheral nervous system, we have our unipolar. So that table on page 394 in your textbook gives you a nice overview of the different types of neurons, where I might find them in the body. continuation of the same, just giving us information about where I might find these guys in the body. So we have sensory neurons, brings information in. Motor neurons bring information out. And then there's an in-between guy that sort of hands off and helps to process information. We call these guys interneurons. And these are the three pathways that we find information passing in the nervous system, both central nervous system and peripheral nervous system. Many of the neurons that we find in the peripheral nervous system are going to be mainly motor or sensory. The integration part we typically find more of in the spinal cord and in the brain. When we talk about the brain, we're going to talk about different areas of the brain and what goes on in those different areas of the brain. And there is going to be a whole group of things we talk about called integration centers. And they are going to connect sensory and motor input and output. Okay? So, the next thing the book talks about is something called membrane potential. And this is the chemistry part, oh yes, there's chemistry even now, of how a nerve impulse takes place. There are chemical changes that occur with the help of these big purple blobs. What are these big purple blobs? They're integral proteins. And what were they for? What were those proteins for when we talked about cells? Yeah, they're going to help with transport of substances into and out of cells. And substances, in the case of a membrane potential, are ions that are going to help the membrane at rest possess what charge? Remember I said we were going to talk about this again, and it was going to be important? What's the charge associated with a membrane at rest? I'll give you a hint. It's polar. Anybody? Negative where? Switch. Positive outside, negative on the inside. That's a membrane at rest. Now, in order for us to change a neuron resting to a neuron firing off and sending information, what do you think we have to do? Yeah, we're gonna mess with that charge. We mess with that charge, and that's how we pass on information in a neuron. There's two sets of very important proteins that are involved in a nerve impulse. One of them, or sets of them, are called chemically gated ion channels. And they are going to change what they do for a living, be stimulated by what? If they're chemically gated channels, what do you think they're going to be changed by? Some kind of chemical, yes? What do you think that chemical might be? Neurotransmitters. Ah, here's where those guys come in. Now these guys happen to be in charge of sodium and potassium levels on the inside and outside of cells. And if they change what they do for a living, do you know what they do for a living normally? These little protein blobs. What do they do for a living? I'll give you a hint. It's a process called active transport. be responsible for keeping something or pushing something against its concentration gradient? What might that be? Sodium. So they, normally, what they do for a living is push sodium against its concentration gradient out of the cell. That causes resting membrane potential. Putting more positive where? Outside the cell. This is the reason why resting membrane potential is what it is. There's other ions involved with these little protein pumps, but that's going to be the major one. So they talk about basic principles of electricity, and basically we're moving charged particles through space. And that's what electricity basically is. Now, they go into some definitions on voltage and resistance and current, and they give us some nice little calculations on what current is based on voltage and resistance. Am I going to make you know those? No. But I do want you to understand that that's what electricity is. So a change in charge being passed along a membrane based on rules of current and voltage and resistance is what is going on in our neurons. And these guys, these membrane channels, are the guys responsible for allowing that passage of charge down these extensions or fibers or axons. Yes?
So when do the chemical gradient and the electric gradient, do those ever counteract each other if they're opposite, if they're going two different ways?
They actually, one sets the other off. Okay? And we'll see that in a second when we start talking about how those ions are passed through the cell. So it's important to understand that that's what electricity is. And these guys, both chemically gated ion channels and voltage gated ion channels, are going to be responsible for passing these electrons down any given neuron. So on page 396, we see described under the role of membrane ion channels, these guys here. They talk about chemically gated channels. Again, some sort of chemical is going to stop them from doing what they normally do. Pushing sodium where? Out of the cell, against its concentration gradient. Now you understand the active transport part, right? In order for me to do that, I need what? I need energy to do that. Voltage-gated channels are going to stop what they do for a living when there's a change in what? In voltage or a change in charge. These guys are going to work hand in hand in the nerve impulse and action potential of any given neuron. And then they also mention things called mechanically gated channels. They respond to some sort of physical change. So pressure receptors in your fingers, for example, are good examples of mechanically gated channels. When we talk about the special senses, we're going to see that there's mechanically gated channels in your inner ear. So when sound waves come in, they cause a change in pressure, and that's what's going to fire those guys off. We also have photoreceptors that are going to be stimulated by light. We have receptors on our tongue and in our nose that are going to be, they're chemically gated channels, but they are going to be fired off by changes in the chemistry of our saliva and our mucus in our nasal membranes. So that's what's going to be the stimulus for those guys. So understand that each neuron has its own type of stimulus. And that's, again, what kind of neuron? Sensory neurons, bringing information in. The other guys, all the guys in between, are going to be fired off by the neuron ahead of it. So those neurotransmitters after the stimulus begins. And this is kind of like if I set up dominoes and I start here and I set a whole bunch of dominoes up. The stimulus for the dominoes all falling, the first one is going to be what? It's going to be me pushing it. But all the other ones firing off are going to be because of what? The domino ahead of it, pushing it down. So that's kind of hopefully, an analogy you can understand. So once I start that chain of change going, it's going to continue on just like a row of dominoes. So, resting membrane potential is achieved by these guys, these membrane proteins that are working very hard and working against the concentration gradient to achieve membrane potential. And remember, membrane potential is more positive on the outside, resting membrane potential, more positive on the outside and more negative on the inside. It's not just because of the ions that we have the more negative on the inside. We also have some very large proteins that sit along the inner surface that contribute to that negative charge. So we become more positive when positive ions rush in. And this is what's going to happen when we disrupt these membrane proteins, whether they be a chemical disruption or a voltage-gated disruption. So, sodium and potassium pumps, or ATPases, are what we're going to talk about when we talk about these guys and membrane potential. So if I took a voltage meter and measured the charge of cells, if I were to measure the inside of the cell, I would get about, and this is an average number, about minus 70 millivolts. And that would be the inside charge of a cell at rest. So keep that in mind because that's important when we talk about an action potential. If you look at page 398 in your textbook, you see this diagram. And you see the important ion channels responsible for maintaining resting membrane potential. So we see our sodium-potassium pump. We see the concentrations of sodium and potassium inside and outside cells. Sodium is at a higher concentration on the outside of cells than it is on the inside of cells. Potassium is at a higher concentration inside cells than it is outside cells. You get a little leakage of potassium through some potassium channels, but it gets sucked right back in by other channels. So, yeah, and it's constantly doing that. So now, sodium channels, we have some leaking on the inside, but they get pushed out by that sodium-potassium pump. So these guys, potassium, sodium channels, and then the sodium-potassium pump, are constantly working to maintain resting membrane potential by pumping ions in or out against their concentration gradient. And these are the guys that help keep membrane potential, resting membrane potential. So what happens when a change occurs? When I look at the voltage inside cells, I can actually measure this. Has anybody ever seen a tracing of this electrical potential? I bet you probably all have and you don't even know it. Anybody ever see an EKG tracing? What is that? No? Yeah, it's the electrical system of your heart. And EEG can measure electrical stimuli in your brain. It doesn't usually look like this though, does it? Okay, we're going to see that in 1 second, what you probably saw when you looked at that EKG tracing. But what we see during any resting time in a cell is it's not always going to stay at minus 70 millivolts, because those pumps are constantly pumping in and pumping out, and some's leaking in and some's leaking out of sodium and potassium. So we get a variation around that minus 70 millivolts at any given time. When the variation of the charge reaches a more positive, which it's doing here, we call that depolarization. When the charge is less than that minus 70 millivolts, we call it hyperpolarization. And it's going to constantly vary in any resting membrane around that minus 70. Always a little depolarization and a little hyperpolarization. So when we talk about those small changes, they're described in your textbook under graded potentials. In order to actually fire off or knock down those dominoes or have an action potential in a neuron take place, we have to reach a certain point with respect to charge. These little graded potentials aren't going to add up to a nerve impulse unless they reach this certain point. So when we talk about a nerve impulse, we talk about something called an action potential. And this is where I'm going to now change this charge enough to actually spread it down the axons and cause changes at the end of the axons. So depolarization, that working towards a more positive, and you understand what depolarization is? If I become depolarized, I'm no longer what? Polar. Very good. So it can happen in small patches. Which means what? You tell me. Change in charge. If I'm polar, what am I? No, I'd have two opposite poles, a positive and a negative end. If I'm depolarized, I'm working towards having a more equal charge inside and outside. So the inside, that more negative charge, is going to become more positive during the depolarization process. Yes? In order for a neuron to fire off, we have to have that depolarization spread all the way down the axon. And when depolarization happens, an action potential will happen. And that is a spreading of electricity from the beginning to the end of a neuron. This is an action potential tracing. So we can see a huge difference in charge when I have created enough change to cause an action potential to take place. When I'm at rest and I'm a cell, and again, I'm measuring my charge on the inside of that cell, I'm at minus 70 millivolts. Depolarization and hyperpolarization is happening all the time. But something changed here. What changed? Depolarization went high enough to reach something called threshold. There was enough change in the cell, enough influx of sodium to bring it up to what we call its threshold point. In this particular cell, the threshold point would be minus 55 millivolts. And when we look at any given neuron, there's a certain area where that change in charge has to reach. And it's right at the beginning of the cell body meeting the axon. That area is called the axon hillock. And this change in charge has to reach that point in order for this whole action potential to take place. So back to my dominoes on the table. If I just walk over to the dominoes and go, and I don't wiggle that first one enough, is that going to cause all the rest of them to fall down? No. So I have to give enough of a change to that first domino enough of a change to cause it to fire off. I have to cause enough of a change inside a cell to reach its threshold at the axon hillock, minus 55 millivolts, to fire it off, to open up all the floodgates so that more of that sodium can rush in and change or depolarize that fiber all the way down. Once I cause enough of a change chemically to reach threshold, guess which channels are going to kick in and cause the rest of the change to happen? Remember, I had two different channels. I had a chemically gated channel and I had a voltage gated channel. Guess who's going to fire off once I reach the right voltage? The voltage gated channels. Guess where the voltage gated channels live? In the axon, exactly. And they begin at the axon hillock. You got it. So I have to cause a change within that cell somehow so that the voltage gated channels will fire off, and I have to reach minus 55 millivolts at the axon hillock to cause a great deal of change to then pass that message on. So this is an action potential. And when the voltage gated channels kick in, they're all going to open up in series and cause a huge change in charge inside the cell. So we reach about positive 30 millivolts. Big change, right? Because of the influx of what? Massive influx of sodium. No neurotransmitters now. The neurotransmitters are the guys that cause the change here. Okay? Now it's the voltage-gated channels that are going to cause the change down the axon. And that voltage-gated change is going to force out, at the end, axon terminals, that neurotransmitter, that chemical which started the whole business in the first place. It's going to start the whole business with someone else down the line. Either another neuron or what are the things that neurons communicate with? Other neurons, muscles, glands. Yeah, endocrine system.
So is that called a negative feedback mechanism then? Because neurotransmitters start it and then get the signal to end it?
No, they don't get signal to end it either. They're just going to keep going. Can I somehow stop those dominoes with my mind as they're falling? No. Once they start, they're going to go. Once a neuron's fired off, it's going to go to the end. You can't stop it. Unless there's a problem with it or a blockage, then it'll stop it. So this is an action potential. We're going to describe an action potential by looking at what's going on within the cell. This little diagram down here talks about the permeability of some different ions. We see that when the full-blown charge change happens, the sodium permeability, and do you understand what that means? The sodium rushing in. It's going to rush in normally because what? Not because the inside's negative, it's because it wants to go with its concentration gradient. And normally, at minus 70 millivolts, I was stopping it from doing that, wasn't I? The chemically gated channels were pushing sodium against its concentration gradient so that we have a much higher concentration of sodium on the outside than we do on the inside. When those guys get paralyzed, and that's how I describe it, by neurotransmitters, they kind of open up. Where's sodium going to go? It's going to go with its concentration gradient. It's going to rush from a high concentration to a low concentration. So the permeability of sodium increases a great deal. Those potassium, leaky guys, they change a little bit, but not enough to change the charge as much as we see the influx of sodium doing. What happens when repolarization takes place? What do you think happens to those little pumps? Yeah, they wake up after a short period of time, and they're going to start doing their job again. They're going to push sodium back out against its concentration gradient, and that's going to cause the charge to come back down. They're doing that so rapidly that sometimes the charge, pretty much all the time, the charge goes actually a little bit lower than minus 70 millivolts until everybody equals out again. And that's called what? Hyperpolarization. So, to generate an action potential, we have to have some sort of change occurring in the beginning to those pumps. If you look on page 400 in your textbook and 401, we see a description of generation of an action potential. So again, some of the events that are occurring involve these pumps, these protein pumps. We have sodium and potassium channels. And if you look at the diagram, you can see the picture of the action potential. And you can see what's going on with these pumps during that portion of the action potential. So at rest, sodium and potassium pumps are working, pushing potassium in, pushing sodium out, giving us that minus 70 millivolt charge. Something causes a change on this end, causing what to rush in? Sodium. And guess who's going to cause that change to occur? Guess who's going to come in contact with the receptor and paralyze it? Neurotransmitter. Or some other what? Some other chemical or some other stimulus. Yes. Okay? Right now, let's stick to neurotransmitters because that's easy to understand. Chemical. Chemical comes in contact with this. Is going to paralyze it. Open the gates. Who's going to rush in? Sodium. Potassium's kind of doing its thing. It's going to get talked about over there during repolarization. But look what happens to the charge inside the cell. It starts to come higher and higher and higher. Eventually, everybody wakes up again, and what happens? Repolarization. The charge starts to come down, the sodium is being pushed back out, and we're going to plug it up so it can't come back in, and potassium is going to be taken care of by the potassium channel until we reach hyperpolarization, and then everybody equals out and does the things that they were supposed to do, just like in the resting stage. Yes, sir? No, dendrite's role is basically the chemical gated channels. So graded potentials are going to add up to changes in charge at the axon hillock. So when I talk about the chemically gated channels that I talked about, those are all going to be located in the plasma membrane portion of the dendrites. So the dendrites is kind of the receiving end of message. So say, for example, you whack yourself in the thumb. That mechanical stress is going to fire off, cause an action potential to occur in the pain receptor or the pressure receptor, that neuron in the beginning of my dominoes falling. That domino, that first receptor, is going to cause a change in charge because of the mechanical stress to cause enough of a change in charge, influx of sodium, to reach the axon hillock of that cell. Once that change in charge has reached the axon hillock because of you whacking your thumb, you still with me? Okay. That change in charge, that influx of sodium is going to flow through that cell body and it's going to hit the axon hillock. Once it hits the axon hillock, what's going to happen? all the dominoes are going to fall. All of those voltage-gated channels, because I reached their threshold, are going to fly open. Sodium's going to rush in, pass that change in charge to the end of that cell, and what's going to be forced out the end of that cell? Chemicals, neurotransmitters. They're going to then communicate with the next guy in line, and the next guy in line, and the next guy in line, until they reach your spinal cord. That message is then going to get handed off from interneurons up to the brain. That information is going to be received, and you're going to say, ouch. That information is then going to be passed down through a series of neurons to the motor neurons. That information is going to come back down. We actually don't pass it up to the brain. We actually cause a reaction before we even realize it happened in that case. That's called the reflex. Don't want to confuse you. But motor it doesn't bypass the brain. There's two roads. So it goes to the brain and also immediately goes motor out to cause you to react. Pull your hand away. Do you ever do that, burn your hand or bang your hand? You have moved. Your action has taken place before you even realize you did it. That's called the reflex. So there's a separate little road in something like that that will allow you to have a motor response very quickly so you don't cause damage. You realize it after. We'll talk about that when we talk about the peripheral nervous system. So did I answer your question?
Yes.
Okay, so it's a series of change that's going on in a series of cells. Many, many action potentials happening through many, many different series of cells. And what causes the action potential to take place? The beginning, chemically gated channels, or mechanically gated channels, or photo-gated channels in the dendrites. That's going to cause an influx of change in charge. If only a couple of those are fired off, graded potential, I'm not going to have a nerve impulse. I have to have enough change to cause a change where? At threshold, in the cell. So a couple of little firings off aren't going to do it for me. I have to have enough change, influx into the cell at the dendrite level to fire off the voltage-gated channels. The change in charge?
Yes.
Yeah, it's going to happen at the axon hillock. Let me go back and show you the axon hillock before I continue. It's the base of the cell body. I'm going to show you on this one. So I'm going to draw now. I haven't done this I don't do this very good, so be patient with me. So this is my neuron, correct? Where are my chemically gated channels? They're on my dendrites. All right, sorry about this. I should use this thing more, but I don't. Shoot. I am challenged today. Pointer options. Come on, pen. There we go. Okay, so when I look at my, where's my pen? Heat this thing. I have to practice more. All right, so where are my voltage-gated channels? Yep, I'm going to make a bunch on my dendrites, yes? Excuse me, these are chemically gated channels. Did I just say voltage? Sorry. These are chemically gated channels. And we're going to find them all over the dendrites. Okay? Now remember, these guys are sitting in fluid, correct? Who's on the other side? I know I want to touch off. I want to touch off. Who's on the other side? Who's at the beginning of this dendrite? There's another axon. There's another end of an axon. Over here. So there's axons over here, the ends of axons. We call them axon terminals, yes? Why is this doing that? It's like opposite. There we go. So axon terminals here. I know this is a terrible drawing of another neuron that just went through an action potential. What comes out the end of these axons? Neurotransmitter. So the little dots I'm making are being secreted by a previous axon. Those little dots, those neurotransmitters, are going to come in contact with those chemically gated channels in the dendrites. Now, if I secrete enough neurotransmitter, I'm going to come in contact with all those little chemically gated channels, correct? And open up all those sodium channels. Sodium is going to rush in and diffuse from a high concentration to a low concentration, increasing the charge inside this cell. It has to be enough sodium rushing in to increase the charge inside this cell at this point, and see where I'm pointing? What is this called? It's the axon hillock. To reach what voltage? Minus 55 millivolts. Once we reach minus 55 millivolts, there's a whole bunch of these channels that we're going to find here. And what are these guys called? These are the voltage-gated channels. They're all going to fly open as the voltage change occurs down the axon. And because it's an insulated axon, that voltage change is going to happen very quickly. That change in voltage is going to be passed very quickly down this cell until it reaches what? More axon terminals. When that change in voltage reaches here, what's going to happen? More neurotransmitter is going to be released. Yes? What's going to happen to the next guy in line? These are the dendrites of that next guy in line. I know, bad drawing. Yes? You getting the point? Until it reaches its destination. So if this is sensory information, all of these are going to be passing that sensory information to the central nervous system for processing. We're going to have a connector in there, those interneurons that are going to connect sensory information after processing takes place to motor information, which is then going to bring the message out to a destination like a muscle or a gland. Yes? Yes. Eventually. When we talk about reflexes, and we'll talk about that in a couple of chapters, there are separate little pathways that information follows. So when I do that, you react before you even realize what I did. When you touch the stove and it's hot, you pull your hand away before you realize the stove is hot. That's called a reflex. So when we talk about the pathways of information, peripheral, central, back out again, we're going to talk about some pathways that are there in addition to help protect you from damage. And that's what a reflex is. Sir?
The reflex in the central nervous system, is that the gray matter or the white matter?
No, it's interneurons, gray and white matter is involved. And when we talk about that movement, we're going to talk about the spinal cord. All right, now this thing's going to screw me up. No, 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 no. There we go. So you with me so far? That question, did I answer that question? All right. Let's go back.
When the neuron is at that point where it just received a message, can it receive more than one, or does it have to repolarize before it can get a message?
Exactly. Once the changes happen, once it's in full depolarization and it's starting to repolarize, there's a certain point in which you can't receive any more message. You have to even out your chemistry first. We're going to see the same thing happening when we talk about muscle contraction. Yeah, they fire off, but think of the dominoes. I got to set them back up before I can knock them down again. And that's what my membrane receptors are basically going to do, set them all back up so they can fire off again. So you can't just keep firing them off.
Sensory, is there a reason why some people have certain neurons and other people don't?
It's sensitivity. So think about different people's neurons. Some people can smell, for example, something way before other people do. Their neurons fire off faster than other people's neurons with respect to that sense. Same thing with pain. So, does that make sense?
Yeah, it does. I just
So people have different sensitivities. Their neurons have different sensitivities to stimulus. Absolutely. And some people can tolerate more pain than others because their pain receptors have different sensitivity levels.
just wondered if it's a negative thing.
It can be a very negative thing. As a matter of fact, some people can't feel pain, and that's a huge negative thing because they're causing damage to themselves and they don't even know it. So the pain and your response to pain is there for a reason. It's there to protect you from damage.
Biological.
Your guess is as good as mine. But it's basically the intensity in which stimulus fires off your neurons. That's different for all people. Okay? So, key players, voltage-gated channels. When we look at focus on action potential, page 402 and 403 in your book, we see all of these guys and how they're involved in an action potential. Remember, action potential involves permeability of some key ions, sodium and potassium, and the changes in that permeability in the plasma membrane associated with the action potential tracing. We're firing things off or depolarizing at number 2, and we're repolarizing, bringing things back to normal with respect to permeability of these ions during repolarization. Again, we kind of overcompensate and we have hyperpolarization at the end. And when an ion or when a neuron is in hyperpolarization, it's going to take more stimulus to fire it off. When you're in a hyperpolarized state, it's going to take more influx of sodium for you to reach what? Threshold. So remember, when you fire something off, it's going to take a little bit more stimulus to fire it off again very quickly because of the fact that you hyperpolarize the plasma membranes with respect to permeability of sodium and potassium ions. So you can see the key players on page 403.
That's why we put it in saline solution.
Put what in saline solution?
Sodium.
Why do we put sodium in saline solution? Not because of nerve impulses.
Well, really,
Really? Why?
to keep the balance, right?
Not because of nerve impulses.
Because you need it, right? You need that sodium.
Think about tonicity.
Well, right.
You need sodium inside and outside cell, right? I put sodium in IV solutions because I want to keep the tonicity of my cells, not because of nerve impulses. Is a sodium imbalance going to mess up your nerve impulses? Maybe. But I'm more concerned with cell tonicity, movement across that membrane. And movement of what? Why do I put sodium in IV bags? Movement of what? Water. If I mess up the sodium concentration, do you see what I mean? Okay. So, these are the events. Those are the players. And again, when we look at the events, we can trace them out because there's a change in electrical charge. We can actually measure that with a voltmeter. So, action potential takes place. Minus 70 millivolts reaches that minus 55 state. We're going to have a change in charge that passes all the way down the axon and takes about how long? 4 milliseconds. What's a millisecond? 4 1,000ths of a second. Happens very fast. Yes? Not just one neuron. When I do that, how many neurons? We're talking about the one that receives the stimulus in my inner ear, then it passes it down to central nervous system for processing in the brain, and you have a result. That's what happens in that time. Exactly, your blink. So not only did we get sensory information, it was integrated, it was processed, and motor information was sent out, all in that same time. So when we talked about trying to re-stimulate neurons too quickly, we can't do that too often. We'll reach a point where the neuron's just going to stay open because it's being stimulated too much. Kind of like tetanus, when your muscles all contract because you're stimulating them too much. When we stimulate consecutively, we're going to notice that many other neurons can be incorporated into that stimulus as well. So remember the dendrites with all of those little extensions? If I keep stimulating a bunch of neurons ahead of it, I'm going to cause that neuron to continue to fire off at a rapid rate, but I can only reach a certain point. The dominoes can only fall so fast, correct? So by stimulating a neuron over and over again, it's not going to necessarily make it a stronger result. Understand what this means. Depolarization, repolarization, hyperpolarization. Understand that during depolarization, sodium rushes in. During repolarization, we get everybody evened out, and potassium is going to leave a little bit, and it's going to come back in. And during hyperpolarization, all the chemistry is evening out. So why is it that we need myelination? And what happens when my axons are larger or they're smaller? Or what happens if they're not myelinated? Well, they don't conduct less, but the movement of message sort of gets diminished. Okay, so bare plasma membranes, the voltage is going to decay, and it's not going to travel very far. It leaks out. In non-myelinated axons, the message is going to be conducted much slower because it's going to slow down and then speed up, and then slow down and then speed up. In a myelinated axon, that's not going to happen. We're not going to have the slow down because of the insulation. So in a myelinated axon, we're going to conduct that message much faster because we won't leak out message as we go by the insulated portion of the axon. So when I look at those voltage-gated channels, where am I going to find them? They're in the nodes of Ranvier. And if I have a myelin sheath, I'm going to pass that message very quickly to the next group of nodes. I won't diminish the change in charge because of the insulation. Sometimes, and I have an older movie I used to show to my class many, many years ago. They described the firing off of the voltage-gated channels in the axons as taking a ball and throwing it to the next person in line. So people had the impression that that message jumped from one node to the next, but it doesn't. It goes inside the axon from one node to the next. Does that make sense? So we don't lose that charge because of the myelin sheath. And that's going to allow this message to be sent very, very quickly. So myelinated axons can send message much more quickly than non-myelinated axons. The other thing that's going to affect how fast things move through an axon is the axon diameter. So on page 405 in your textbook, you see the discussion about conduction of velocity. So the larger the axon diameter, the faster it conducts an impulse. How come? Yeah, less what? Less resistance. Degrees of myelination are also going to affect how fast message is sent. So if we have a highly myelinated group of fibers that are receiving and sending this message, they're going to go a lot faster than if we have a group of non-myelinated fibers. So the degree of myelination of fibers is also going to affect the rate at which messages are transmitted. To talk about homeostatic imbalances on page 405, what is multiple sclerosis? It's described as something called a demyelinating disease. There's a whole bunch of other ones. But not necessarily the brain, but in the peripheral nervous system too. So when you demyelinate these neurons, what's going to happen to message that's being sent to its destination? It slows down, and eventually might never even get there. So we're talking about muscle contraction and coordinated movements when we talk about the nervous system and how it communicates with the peripheral end of the nervous system. So demyelinating disease can reduce the message and the rate to the point that they don't ever reach their destination. Nerve fibers may also be classified according to their diameter and the degree of myelination. So we have group A fibers. Those are somatic, sensory, and motor fibers. What's somatic, sensory? Sensory, or it's involuntary in that peripheral, your body. Okay? Then we have group B fibers. They're lightly myelinated. They're kind of the intermediates. They have an average rate of travel. And then we have group C fibers, smallest diameter, non-myelinated, much slower pace. All of these guys, B and C, include autonomic nerves. What's an autonomic nerve? Things that automatically happen. Things that innervate, and that's me getting my message to where it needs to go. Visceral organs, visceral sensory, visceral. What's visceral?
Your organs.
Yeah, internal organs. Okay? Synapses. What's a synapse? No, it's actually
That's
the...
where the chemical reactions take place.
Here's an axon. Here's its end. Yes? There's a synapse between the end of this guy and the beginning of this guy. A space. Yeah, a physical space. That's filled with what? Fluid. There's fluid all over the place around our cells. These aren't actually attached to each other. There's a space between them. It's very, very tiny, but that space is called a synapse. So, there's a whole bunch of terms with respect to the synapse, the space between one axon and the next group of dendrites, for example. So, we can have something called an axodendritic synapse. What's that? Between the axon and the dendrites of the next neuron in line. Axosomatic synapses. Soma means what? Body. Does a neuron have a body? It's cell body, right? So this is a synapse between the axon and the cell body. Are those going to fire off the next neuron in line a little bit faster than an axodendritic synapse? Yes, because it's going to cause a change in charge closer to the what? To the axon hillock. Presynaptic neurons are neurons before the space. Yes? What's a postsynaptic neuron? A neuron after the space. And all of these guys are communicating with each other to send message. So we can have different types of synapses that'll send different types of messages across the space. We can have electrical synapses. So there's actually a change in voltage that passes from one to the next. The one we've been talking a whole heck of a lot about today is a chemical synapse. So we see chemicals passing from the axon of one through the space to the dendrites or whatever we happen to be talking about of the next neuron in line. So chemical synapses, spaces, are going to have chemicals that diffuse across the space and affect these plasma membrane receptors on the other side. What are the chemicals I'm talking about? No.
Salt.
No.
Sodium.
No. Neurotransmitters. Now, sodium and calcium are going to help this cell squeeze out the little packages of neurotransmitters into the synapse. Sodium and calcium are going to help this occur. But it's these little chemicals in these packages, the neurotransmitters, that are going to cause the effect in the postsynaptic neuron. They're created in the soma, in the body, and then they are transmitted down. A lot of those glial cells are going to help bring substances to the cell so they can create their neurotransmitter. But they're stored in the axon terminals. All right. What do we got left? Say it again. I'm sorry, hon. The original stimulus. No, the original receiver of information, if we want to give one to this picture, would be way up there, the presynaptic neuron. So you're banging your thumb, the pain receptor, that stimulus that fired off that neuron, this would be the end result. So this is the end of that first neuron. That neuron is then going to release exactly into the synapse. The reason it's going to release the neurotransmitters into the synapse is because there was a chemical change that took place within the cell. The combination of chemically gated and voltage gated channels fired off that neuron. What caused it to start? That pain. And that's the beginning of the message. This will then send the message to the next and the next and the next. Okay? So I really feel like I want to give you a quiz on Thursday. I really do. The only reason I really do is because I don't want you to go sit back on your haunches and not study this. This is very, very important information. This information is very important for you to understand so you understand the rest of the chapters. So I'm going to be the nasty, rotten witch that I am and give you a quiz on Thursday, chapter 11, the whole chapter. About attendance. Oh yeah, get your clickers out. Let's ask a question.
Like, so the stuff we didn't cover as well?
Correct. The question was the stuff we didn't cover as well. The answer, yes. I want you to study the whole chapter. Now, did you know that you have a lovely little tool that's going to help you study this chapter? And today, you get to crack it open. It's called interactive physiology. And you were wondering when you were going to use that, right? You're going to use it today. Because you're going to go in and you're going to pull out the nervous system module of interactive physiology. And you're going to look at action potential and membrane potential. And you're going to watch that little movie and you are going to be enlightened totally.
Is that on our DVD? Yeah.
It's on the DVD, but it's also in mastering. So don't panic. You go into mastering, pick the textbook. It's right there too. Okay? So let's have one little questione before we go so we can take attendance. I am definitely spatially challenged. So, the nervous system has 3 overlapping functions. Which of the following represents a logical sequence of the 3 functions? That means they have to go in order. Maybe. I'll sneak one more in. So they got to be in order. Pick. 5, 4. Just make sure you click, no matter what. 3, 2, 1. Only 43 people? We have scared way too many people away. What's the answer? The answer is C. Good. All right, let's do one more. Sit down. Holy moly. I got one more minute. All right, while studying for an exam, you reach for a beverage. To extend your arm, your nervous system is activating what? Five, 4, 3, 2. What's the answer? Somatic. All right. Go study chapter 11, quiz on chapter 11 on Thursday. We'll finish it and start chapter 12.